Hi everybody, we're back and we are on to lecture 9, Analysis of Variance, ANOVA analysis. And let me change my pen, even though I like cyan, let's use our red. ANOVA analysis. So you've probably heard of ANOVA analysis, hopefully in some engineering context, probably in a lot of biological contexts. And ANOVA is an extremely useful statistical and analytical tool. So it's really, really useful when you have multiple variables and you're studying them simultaneously and you want to determine whether they're going to have an effect on the measured outcome of an experiment. So, for example, if I have a high-strength alloy, I want to create some new alloy to maximize tensile strength. I can vary some parameters. And one of the questions you may have is, like, if I have multiple variables like annealing time, solute concentration, work hardening, are all these variables, or are any of these variables, do they have a statistically significant effect on your UTS? Which parameter has the largest effect? Do any of these factors have a combined effect where they're functions of each other? They're not, they're correlated parameters, right? Even if it is statistically significant, does it have a large effect? So these are just some of the questions that we can answer using ANOVA. And we're going to start off simple, and we're going to use essentially a one-way ANOVA analysis. But before we get into a one-way ANOVA, let's look at, we're just going to use kind of a play data set. So specifically, we're going to use this data set called crop data. And in the crop data, we're going to vary density. So we're going to have a density with parameter 1, parameter 2, low, high density. We're going to have a parameter where we're looking at the yield of crops. So I could do high density crops, or excuse me, low density crop, high density crops. I can use or plot essentially, or plant, excuse me, not plot. R's on my brain. I could plant the crops in block 1, 2, 3, or 4. I can also give them fertilizer type 1, fertilizer type 2, fertilizer type 3. So you can see here, I'm varying these variables in each of my experiments, and I'm measuring an outcome, which is the yield. So we want to determine, again, do any of these variables have an effect on the outcome? And ideally, we want to build up towards, we want to look at each of these and see which has the largest effect, and is it a large effect, and what parameters are significant. So you can extend this significantly. So anytime you have an experiment where you're varying multiple parameters and you're measuring a single outcome, you can use this type of ANOVA analysis. And we're going to do this in R. So that's going to be the goal of what we're going to be moving towards as we continue to build our ANOVA analysis. Here we go. My screen was not responding. It was bugged. So next time we're going to start off simple, relatively simple, and we're going to do a one-way ANOVA analysis. So we'll define some critical parameters, but it's really, we're going to be getting in deeply into kind of how do we program this in? How do we determine and analyze the results? And we'll be back next time for that discussion. Thanks. Bye.